So I hope you all enjoyed the show so far. It has been a really good interaction with suppliers, equipment manufacturer, and farmer and users like us. So today I'm going to talk a little bit about like the cell therapy and automation. I think a lot of the panel discussion happened earlier today. Also a few presenters kind of mentioned the importance of automation. And where we want to achieve for decentralized manufacturing, automation is also a key to that as well, right? Yesterday in the keynote panel discussion, a lot of good discussion about how to drive down the cost and to make it more accessible for patients. Because another day, we're manufacturing a therapy that can revolutionize cancer treatment, but you also want to make it affordable so they can treat more patients. So disclaimer, right? I'm not really an automation engineer or specialist. I'm more like a material scientist by training. I got a degree in polymer engineering. So, but I work closely with a lot of automation technology evaluation, right? So this is really my observation about Kite's journey in the technology evaluation and in implementation into manufacturing. What are the challenges we faced in the past? So if you're going along the journey about automation, technology evaluation, so these are things you should consider, right? First, a few corporate slides, like normal. So we are a single-focused cancer cell therapy company, right? We have a few products approved on the market, but cancer cell therapy is really CAR T cell therapy is what we only do. This is our background slides about how we get here so far, right? I think one of the panelists mentioned yesterday, right? 15 years ago, this cell therapy is kind of a science fiction, right? I mean, everyone thinks it can revolutionize cancer treatment, but so today, really where we are here is we actually achieve a lot of the several CAR T products achieve the blockbuster status, right? Back in 2009, Kite was founded as a clinical stage startup, right? Then we work with the NCI, try to bring this anti-CD19 CAR T cell therapy. Then 2017, we were acquired by Gilead. Same year, we got first Kite product approved by FDA, Yescarta. Then 2020, we got a second product on the market, which make us, at that time, the first company to have 2 commercial products on the market, right? So really, I want to point out this number here, 31,000 patients by last month. See if the pointer is working, right? That's really like, I think it's really a big achievement for the CAR T, not only for Kite, it's really for the CAR T cell therapy business, right? So before, like 10 years ago, no one really imagined this, right? Because there's a lot of challenges in the manufacturing of autologous cell therapy, right? Because the manufacturing process is so long, it's very tedious, really why we need automation, right? How automation can help us to get even more patients. I think one of the panelists also mentioned that only 2 out of 10 patients really only have access to CAR T at this moment. So with so many, so this is kind of a little bit numbers by different things. So we have 2 products that kind of approved for 5 different indications. Because the unique challenges of autologous cell therapy, you literally manufacture 1 lot for each patient. So you need a lot of space because Amy mentioned about the scale out versus scale up, right? This is not something you can scale up to 1,000 liter, 10,000 liter, then manufacture on batch, right? So you need a lot of clean room space. So a lot of upfront investment on your clean room facility and also your manufacturing, right? You have to train these operators to do all these operations manually. Really, that's why we have like about over 4,000 employees globally, serve 30 countries. So cell therapy, just because it's unique challenges in the manufacturing, in the patient, you have to really make sure you can achieve a high success rate in your manufacturing. So it doesn't require a high specialized team. That's why when Gilead acquired Kite, make us still stand alone units, really just work on cell therapy. A little bit about our global presence. So both myself and Mahesh and a few other colleagues come from our Santa Monica headquarters, which is where corporate and also our global R&D center are located. We also have a research site at Foster City, co-located with the Gilead research group. Our first commercialized site is in El Segundo, which is about 10 miles away from Santa Monica, but in normal LA traffic, it usually takes you about 1 hour or so. We also have in-house vector manufacturing located in Oceanside. We are one of the few companies who have fully integrated viral vector in-house manufacturing and T-cell manufacturing. Because viral vector is such a critical raw material for us, there are a lot of supply issues in the past, which we made a decision to bring the viral vector manufacturing in-house. We also have a European manufacturing site located in Holland to serve the EU patients population. Then Maryland is our newest addition for commercial manufacturing, where it's running a lot of the automatic process and equipment. So cell therapy is such a unique autologous, for example, it's such a unique manufacturing process, require a lot of close collaboration. That's why our process development and clinical manufacturing and viral vector are both located in California. So the commercial manufacturing really how to you distribute them evenly to serve different regional patients. A little bit about what makes CAR T cell therapy unique, right? I mean, I think this is a slide everyone probably pretty familiar with it, but in traditional biologics, like most of the BPI audience here for, they're talking about like how you scale up, right? You start in the same set of raw materials, you go through different scales and manufacture, and go through a lot of release, and then you can distribute all the shelf to the different patient population, just ship it around. But in autologous CAR T, right? So you're dealing with every single patient, which are at different stage of their cancer. And some of these patients are really at the very, very end of the lifespan, right? You really have only one shot to make it right. And there's also a lot of variation in the donor, in the patient situation, right? Some may be treated by chemo or others treatment before, and their like cells condition is highly variable. So the inherent variability of the starting material, the patient cells, and also a lot of the raw material we use, human or animal derived, there's also variability in there as well. Right? Then you go through a very complex manufacturing process, which we're going to talk a little bit in the next slide. There are a lot of coordination you need to work with the treatment center. When is the right time to draw the cells from the patient, and how you schedule internal manufacturing. That's why we have kind of put a different manufacturing sites locally, right? I mean, as a decentralized panel discussion one, right? The ideal goal is, right, you can have different manufacturing units, equipment, automation equipment in the hospital, but I think we still have a long way to go to get there. So how you coordinate with treatment center with the manufacturing schedule, then eventually manufacture this like individual lot for each patient. This is kind of a high-level, step-by-step manufacturing of our commercial products, right? You'll start with collecting the material from the patient, and then ship to our different manufacturing sites located globally, go through some kind of enrichment and separation to separate the target cell population, then go through some kind of activation process to activate the cells ready for transduction. Then you use the different viral vectors. In our traditional commercial product, we use retroviral vectors. Moving forward, a lot of the shifters are going to lentiviral, right? So there's a difference as well. So then you go through the expansion phase to make sure you achieve the dose level you want, then schedule a ship, freeze it down, formulation fill, freeze it down, ship back to the treatment center, infuse back to the patient. So CAR T autologous manufacturing is such a unique process, so it requires a lot of collaboration with the development, manufacturing, supply chain, and then the whole network to make it happen. I'm not going to do the details, but a lot of the presentation we heard today actually cover a lot of these aspects as well. So this is kind of really like, I think, discussed a lot of times, right? Why we need automation, because just historically, there's so many manual operations. I mean, I used to work in the biologics world as well, right? Where everything has been really end-to-end, fully automatic connected processing, right? You can connect your upstream and downstream in a continuous manner, right? We're talking about continuous manufacturing in traditional biology, but really, when I first came to cell therapy about 5 years ago, I was really surprised. There are so many operations. You literally use a lot of these BSCs, kind of do open operations, transfer samples and reagents in and out to your next step, right? How do you make these more connected? Because every single step here means there's a risk of a leak, a contamination, and you also make sure every single operator you're doing this day to day are highly specialized. It requires so much training up front, right? If you can make these all automatically, you just load on one machine, press a few buttons, let it go, that would be perfect, right? So there's definitely room for automation. So Kite has been really on the frontier, really, automation, right? Because we launched these products, we realized the gaps, and we've been working closely with several different vendors on the market, trying to kind of bring automation into our commercial manufacturing as well. But if you have a commercially licensed product, every single step you want to change will require a lot of negotiation, discussion with the agency and how you implement it. So we have been using different approaches. So we have different process. We have like a semi-automatic process. That way you can start like replacing unit by unit operations with equipment, and instead of doing manually, right? So for example, we have like a filling operations that can be done on semi-automatic equipment. And we also have the isolation enrichment process step done as well by machine. And then we also look at different ways can you really incorporate multiple unit operations into one machine. That's what we call a fully automatic process that can do activation, the transduction gene modification, and expansion all in one machine, right? So just because it was a commercial licensed product, so we have to really take different steps, different approaches to implement this automation into manufacturing. This is kind of just an overview. I think I kind of cover a lot of these topics here, right? Just in the early days, it's very, very manual. And we see a lot of, and as you heard in the presentation earlier, there are a lot of call for automation, really how to make it just easier. And recently, there's also a lot of discussion with AI, how to integrate AI into your process analytics and kind of drive this right forward. So for companies that's kind of like a little bit new to clinical or even commercial manufacturing, right? There's a few factors you should consider, right? For technology adoption, right? Really, clinical success is always the first thing you should consider, right? If there's no clinical success, there's no point in implementing anything, right? So you really think about in the investment, because every time you invest in automation, there's a lot of CapEx upfront, right? You want to make sure this equipment is really used and leveraged, and regulatory support, right? Talk with regulatory, because they always encourage you to how to close the process down instead of doing more open operations. But you have to really figure out if it really helps, right? Doing comparability study to demonstrate your quality, your CQAs are still comparable, right? Figure out your manufacturing challenges, because there's so many different versions of even autologous cell therapy, right? I mean, if you talk about TCR-T versus the traditional CAR-T, it's quite a different process. And if you put allo in as well, every company has their own version of their process. You know your process best, right? Then you work with the suppliers to figure out how to best develop the equipment, the disposable kit to work with your system, right? The cost reduction, right? And a lot of times you really figure out, does it really reduce your cost, right? And how do you connect the different reagents? Because you can make a perfect machine that's running fully closed, but a lot of the incoming material you're working with must still come in vials, right? How do you connect these incoming raw materials with your machine and feed into the automation equipment? These are a few examples. I mean, you can consider for cell therapy process you can improve on. Really, like the folks should really do everything. You try to do everything in a closed manner as much as possible. If there are anything, like I said earlier, if the reagents doesn't come in a closed manner, figure out ways how to better connect it so it can easy to load it. then make sure your product quality control and release testing is still viable. I have 10 minutes left. Let's see. So figure out really your business need, because I think one of the panelists mentioned as well, right? I mean, in his past 30 plus years, no forecast, no supply chain forecast is ever accurate, right? But really, like if you want to deploy a large capital into automation equipment, especially into commercial manufacturing, right? It means like how do you modeling your demand, right? You don't want to unload like so many different patient lots and so many equipment and just sitting idle there. And like for us, right, we have a commercial product. We really need to figure out what our commercial product demand is, right? Is it changing? And also look forward next 2 years, next 3 to 5 years, what will be pipeline need, right? Because RVV process where we run legacy and versus the next pipeline product we're developing based on lentiviral, which is a completely different process. And now everyone talk about the in vivo delivery, right? So the demand will shift dramatically in the next few years to really understand your process, what's coming in your pipeline, and figure out like how do you model these and which will be built into your automation and equipment deployment. Let's see, the material and maintenance of labor part as well, right? Because historically, if you have like a huge population of very skilled operators, right, with these manufacturing improvement or the machine equipment deployment, how do you train these operators to become familiar with the equipment with a completely different skill set, right? And how does the mean impact to your overall employee account as well, right? The easing of integration is also one thing as well. You should consider, right, as the example I give for our commercial implementation, we look into the different unit operation for the semi-automatic and also try to do an end-to-end, right? So think about your process to figure out which way is more applicable to your process, right? Because unit operation is a little bit easier to implement just because you just really compare with the performance of each unit. But if you look into the end-to-end deployment of the platform, which means a lot of your IPC could change as well. How do you control your IPC values and demonstrate eventually your comparability of your process? Supply chain is also a very important topic to consider, right? I mean, because every time you move to automation, which means you'll be locked with that supplier for quite some time, right? And how do you establish the supply chain of these machines and the disposable kit coming with it, right? And also, what is the durability of this technical support, how the relationship comes, right? With tariff coming in, you need to consider where these equipment and the disposable kits are manufactured. Do they have a global footprint to supply your local region and manufacturing facility as well, right? So there are a lot of things you should consider for automation and technology evaluation. So this is kind of like one of the slides one my colleague put together for technology evaluation. So in the ideal case, you will just think, okay, I evaluate the technology in development. Then when I deploy to manufacturing, I can just quickly realize the cost reduction and lower the risk and very, very quickly. But in reality, right, a lot of things work really nicely in PD in small scale. But when you start deploying the manufacturing sites, and especially different regions, you realize that in different locations will have different feedback, operator will have different preference as well, right? How do you kind of realize? And actually, a lot of times it takes much longer than you originally forecast with the cost reduction. And another thing we face is our manual success rate was so good. We're actually one of the industry leaders. We achieved 95 to 96% overall success rate on the manual platform. But then when you try to switch over to automation, how do you make sure you can consistently hit that high bar there, right? I think we only have a few minutes left. Let's just kind of go over. So I think my automation folks also talk about in PD, they also run into a lot of the supplier give them like a closed black box automation equipment, but when you deploy, And there's a lot of push in manufacturing to see how you can open up the system and the architecture as well, right? I'm no automation engineer, so I'll try to skip this slide as quickly as I can. So I got 5 minutes left, so maybe just go to the last slide, which I can speak a little bit more about the material unique challenges. Again, I'm a material scientist by training. So back to this unit operation versus end-to-end equipment design, right? Because how you want to really implement, evaluate this technology, right? Again, unit operation is just like, it's very, very straightforward, right? You know what is your income and outcome for each unit operation. You know your menu setup and requirement, and it's much easier to just say, okay, I want to evaluate this machine here if it achieves the same performance. But as long as you want to like make it more easily end-to-end process that impacts the machine design, right? The more you want to achieve in one machine, and the more complicated it is, the software and the disposal kit, right? Then the end-to-end disposal kit design, right? The more you try to fit in, and this eventually just become like a spider net. It connects so many different pathways. How do you make sure you may have 5 raw material inlets, and you have 3 outlets for different processes as well, right? All these would add this risk, because in your consumable design and what it means for the lot release testing, right? If it's just really a simple bag or filter, it's much easier from the supplier side to do like the QC control and the lot release testing. But if it gets so complicated, it adds a lot of complexity, even challenging for them to do a lot of the QC release testing for endotoxin as well, right? It may even just exceed their previous validation on sterilization as well, right? Do they need to kind of create special sterilization validation for these kits, right? The robustness of the consumables is also a very important thing to consider, right? How do you make sure, because you eventually just load these patient cells into these machines on the same disposal kit for a long really period of time? How do you make sure the robustness is consistently met? And also during your shipping and the packaging validation, we also adds a lot of complexity as well. Particular control, I mean, I think there was a lot of good discussion about like how CGT space particular control is so critical, because it's not like it's traditional biologics. You can add so many 0.2 micron filters everywhere, right? Here is your process, you use a product, right? Anything you introduce into it, it's kind of hard to get it out. So you really need to consider what is your particular control strategy. Now it's just because the kit is so complicated, how do you guarantee all the raw material come into the kit will eventually meet your particular control strategy. Again, the last thing is kind of the last 2 things is really how do you connect to the different reagents will come in vials and make sure that they can be closed in connected in a closed manner, right? Extractable leachable is also something need to be considered, right? Because traditional E&L assessment, you also look at the component by component level. But now, I think agency is calling for more about the cumulative assessment. But if your cells and all your processing is in one kit for, I don't know, a week or a month, right? What it would be the eventual impact on your E&L assessment and the tox assessment you do for the patient, right? So there's a lot of consideration. Again, to sum up, cell therapy manufacturing, especially the autologous one, it's just very, very challenging. So it does require a lot of automation here to make it more easily accessible and lower the cost of those good things. And it calls for really close collaboration between the end users from pharmaceutical manufacturing with the equipment manufacturers and also even the reagent supply as well. How do we work really well together to make this in a closed manner? The consumable design is also something we should consider very early. You don't want to kind of wait until your commercial deployment. With that, thanks for your time. I'm happy to take any questions.
Hi, I'm Therese Choquette from Pharma. Thank you for a really good talk. I enjoyed it, and I think it brings up a really important item, how to transition from the more manual to the automated processes. And I just want to hear your thoughts on what is the most critical step in the whole process of changing from an open to a closed system. Where would you put the most effort?
So I think just like every single step listed here is very critical because you're literally manufactured for that patient. Anything you mess up front will eventually impact your later process as well. But what we really see on the process side is really how do you do the isolation, activation, and transduction. These three are the very, very critical. How do you manage your reagents and equipment, your automation equipment to achieve the performance of the three single steps? Because when start goes in expansion, you really just make sure the cell grew happily with the right level of nutrients, right? So these three steps up front is very critical. But when you go to the formulation field, that's also a little bit tricky as well, because you want to make sure you fill the right volume to meet the right dose level, right? The particular control is also very critical in the formulation field as well.
Yeah.
So different operation steps will have a little bit different unique requirements.
Yeah. Did you find a lot of particles after you switched to a more automated?
I don't have any data here, but like I say, when you switch from like a manual unit operation, which is your particular control can be like isolated to your formulation field. But if you switch to automation, when you consider end-to-end, you combine so many operation steps in one machine, that does add a lot of complexity. So it causes a lot of early on evaluation on particular as well, right?
Yeah. Yeah, thanks a lot. It's complicated because you do risk to change the product with the implementation of new steps. So yeah, thanks for sharing.